All right. Hi, I'm Ian, and welcome to the How Plant Scientists Work series at Plant Biology 2016 in Austin, Texas. I'm here with Sisi from the University of Florida, and she's going to answer a few questions about how she works. So, hi Sisi, tell us a little bit about yourself and your background.
All right. So my name is Sisi, and I'm from University of Florida. I'm a PhD student who will graduate in August, and my study is mainly on guard cell metabolomics and proteomics in response to carbon dioxide. And our organism is Brassicanappus as well as Arabidopsis.
Okay. Okay, good. And so what's one word that describes how you work?
Challenging.
Okay.
Yep.
Good. All right. What's a favorite thing that you do at work?
My favorite thing is, well, learning new things all the time, especially since I'm working with omics data. We need to do a lot of data analysis. I'm a biology background, so essentially I don't know anything about data analysis, but during these 5 years I've kept learning programming and found it quite intriguing. And hopefully I can find a position quite related to this field.
Yeah, I've been learning R lately, and it is kind of fun when it all works,
Yes,
right?
Exactly.
Yep. Okay. So what is your favorite plant?
My favorite plant is still Arabidopsis.
Okay.
Because we got so many resources for it, and just feel like we can explore much more stuffs with Arabidopsis, but still it will be really, really more effective. I mean, if we have more information about all the crops. So.
Okay. Okay, no, that's fine. And you've already told us a little bit about your project, but is there anything more you'd like to say about a project you've been working on lately?
Yeah, so lately I've been trying to confirm several of the new hypotheses that are generated using proteomics analysis with guard cell. There are many phytohormones that we never know that could be responsible to carbon dioxide, and I'm now trying to figure out exactly what they're doing in these processes using both biochemical and genetics approaches, and hopefully we can confirm those findings and find an interesting story. Yep.
Okay, so let's get to the rest of the interview questions, the specific questions. So what are some tools, apps, or websites that you use or visit every day? Do you have a favorite resource?
Yes, my favorite resource is first of all Metapsych and then CAG. Of course, we work with metabolism a lot, and without checking like too much detail into the papers that specifically on specific metabolites, those websites give me a general overview of the whole plant metabolism pathway and as well as genes related to them. It's like quite good tool to start up with. And also it gives all the references linked to each pathway. So I like that a lot. Also, since we work with MassBank a lot, two more databases that we'll be using. First is MassBank, and the other is Respect the database specifically for plant metabolism. And whenever I have a compound that I don't know at all what it is, those are the places I go to find out.
Okay, no, awesome. All right, and so what's an obstacle to doing an experiment that you figured out a solution to?
All right, so originally when I was starting, no one in our lab had been doing carbon dioxide treatment at all, especially when we're working with guard cells specifically. We don't want any interference from other cells.
Right.
So I've read several papers using like a small utility where you can just fix the epidermal peel onto a slice and then monitor the change of their response to carbon dioxide by infusing a buffer like beneath that little epidermal peel on the slide. And that is quite intriguing, but I don't find out where we can buy those kind of instruments. So actually made up a slide by myself using drills and like several pieces of slides stick together, and that works out quite fine, but it still takes a long time. So yep.
But you found a solution to the problem.
Yes,
which
exactly.
Which is awesome. Okay. So last question. So what's some of the best advice you've ever received?
All right. So when I was starting up, I was curious to all kinds of fields, not just for guard cell, and when I was reading paper, I just reach out to all kind of questions that I'm curious about, but that actually takes a long time. And both my advisor and my coworkers tell me that you have to keep focused, like just on your study, because that's where it's gonna take you to places. Knowing all the stuff won't really help if it's not focused on specific field. So that's one of the best piece of advice that I've had. So.
Okay, no, that's good. Yeah, I actually like that a lot. Like I wish I was more focused sometimes. Okay, so the last thing is, if you'd like to nominate someone to do this series of questions next, who would you nominate?
Okay, would it be a student or a
Can be anybody.
Anybody?
Yeah, it can be anybody who's in the plant science world, defined very loosely. So it can be so it can be a professor, it can be another fellow graduate student, it can be a postdoc, it can be an industry scientist you happen to know who's in the plant bio world.
Okay. I would love to see actually one of my committee member. His name is Kevin Foda.
Oh yeah.
Yep,
I know Kevin Foda.
I like to see how he answers these questions.
Okay.
Yep.
Absolutely. I will get in touch with him and put these questions to him.
Great. That would be great.
Probably by email, but all right. Sisi, thank you very much for doing this series.